antibodies are specific for a number of reasons, and we thought that it could address one of the major problems in the field, which is that CB2 small molecules, even ones that were purportedly quite specific for CB2 over CB1 in vitro cell-based assays, once you put them in vivo, you often would still see some sort of crossover reactivity. This is a common thing that's seen with small molecules, particularly once if you dose and you have a sort of a high peak dose, when you first deliver the small molecules, even if it's selective, there's still a high amount of the drug that crossover and cross reacts with the unwanted target. And in this case, it's really important to avoid CB1 because, as you noted, CB1 is sort of like the yin yang partner to CB2, right? So in certain in certain tissues, activation of CB1 actually is pro-inflammatory and pro-fibrotic, right? So you really don't want to have any cross-reactivity between the two because you're just like causing more of the disease and undercutting the effect of your drug for a CB2 activated drug. And the beauty of this too is that where the antibody would bind in comparison to the small molecule is that if you look and compare CB2 to CB1, the small molecule binding sites are super similar. So if you just like align the structures and overlap them, they're hardly distinguishable, which actually, quite a bit of credit to the small molecule developers to find something that could be even, remotely specific between the two. But if you look at the area where the antibody is binding, sort of like on the outside of the cell, on the sort of top loops of this helical bundle of the GPCR, those regions are very divergent. Right? So if you compare CB2 to CB1, it's like 26% sequence identity. So it'd be impossible to find an antibody that binds both, much less an antibody that binds both in a way that activates both. We picked CB2. We're listed this molecule on Bionakes, and we're really excited to be starting to talk to partners about this. There's really interesting use cases in peripheral neuropathy. We have a lot of data in chemotherapy-induced peripheral neuropathy. We're expanding that into diabetic neuropathy now. We also have some good data in liver inflammation and NASH use cases as well. So yeah, really exciting times for that molecule.